So for our reads, the number of total and successful reads was that we needed to choose a different sample rather than the sample that we started with because the samples weren't run, and we chose the samples 307 and 343. The number of total and successful reads of sample 307 was 3,223, and for sample 343 was 2,687. So diversity and relative abundance of bacteria detected by sequencing was that we made a graph for the kingdom, order, and genus, and for every graph of bacterial composition of the fecal samples, we labeled X as an organism detected and Y as percentage. So for kingdom, it shows that the top 2 of 8 classifications.
Go.
For sample 307, there was a high percentage in bacteria and less percentage in unclassified, and for our order graph, it shows that the top 3 of 8 classification was Clostridialis, unclassified, and Bacterialis, which is the classification in the chart. Sample 343 had a high percentage in unclassified than the sample 307, and for our genus graph, it showed that top 3 of 8 classification, Clostridialis, unclassified, and Bacterialis, are the classification in the chart. Sample 343 had a higher percentage in the sample 307, and in sample 307 there had a low percentage of Clostridialis and Bacterialis. I hate you so much. So between our categories of other and unclassified, the other category were reads that could be classified but were present at less than 3.5% of the total reads and thus were not considered important. The unclassified category are ones which do not match any known organisms at the kingdom level and were likely meant that the reads were errors. So discussing our DNA extraction from scat, it wasn't really successful in our group because the terms of quantity were too low, and our DNA concentration must have been between 250 to 260 in order to have enough concentration to track. There was too little or too much DNA, and there was a high likelihood that our DNA was contaminated, and the UV spectrometry and our UV spec results show that. So discussing our amplicon PCR results, the amplicon PCR results were unsuccessful in the gel electrophoresis because as we pipetted our samples into the wells, some of the samples leaked into the neighboring wells and thus it contaminated our results as seen. Our negative control ended up having DNA in the sample because of this, and there were no bands present on our first 3 samples of DNA, and the concentration was too low to detect anything. So discussing our quantification results for the index PCR cleanup, the quantification results from the index PCR had very low concentrations of DNA from all of our samples, and DNA was found in our negative control, and our positive control had our E. coli DNA. We expected to have 0 DNA in our negative control, and we turned out that we did have DNA, which indicated that we somehow cross-contaminated our samples. And discussing our positive control, most of our organisms found in our samples were unidentified organisms, about 51.52%. Bladia hydro... You gotta stop doing that. Bladia hydrotrophica was an identified organism found in our samples, and Bladia was a bacteria found in normal gut microbiomes and eukaryotes to process sugars like lactose or glucose, and even indigestible materials such as plant matter. Another identified organisms that we found in our samples were bacteroids. It made up a big percentage of our findings, and the bacteria is commonly found in human intestines. They assist in breaking down foods, however they are introduced to other parts of the body where infections may occur. And what our results tell us about cross-contamination is that it happens somewhere in the middle of our processing of the DNA concentration, which was a possibility that we didn't follow the instructions as properly as we should and possibly switch the samples on accident since the negative control wasn't supposed to have DNA in it. So discussing the number of successful reads in each of the animals compared, there was no successful reads. Unfortunately, there was no successful reads to amount to the fact that the animals were compared and did analysis on, and there wasn't enough concentration of DNA to get a proper read of our samples. And basically discussing the differences of the gut microbiomes of the groups of animals we chose was that bacteroids were a genus that were found in all of our animal subjects and in different orders. Sample 343 had Bacteroideaceae, and sample 307 had Bacteroidalis. Bacteroids in sample 307 had a majority percentage, and the genus level in sample 343 was 7.32%. In Bacteroids, in the genus of Bacteroides. There was a very small difference between the bacteroids at a genus level, but they were classified into different orders, and we couldn't find details on specific orders of the bacteroids, and our source of this was MicroWiki. So our question was to analyze the gut microbiome of 2 dogs around the age of 10 months or younger of different genders to see whether females had a better gut microbiome than males, which made us hypothesize that female dogs might have had a better gut microbiome. But our data did not support our hypothesis because our results were skewed and our DNA concentration was too low to gather adequate results to support or deny our hypothesis. Thank you.